This presentation made by Mariska Oberg and I, Shirley Jacob, will be on the Antibiosis Lab. Data will be taken from the Spring 2021 Microbiology 1 Notebook, Lab Section 2, Section 3 Northeast, 3 South, and 4 North. The image on the right shows an example of what we are trying to achieve with this lab. The isolates with a clear ring around it will be considered successful as they have inhibited the bacteria around it.
Certain bacteria possesses the ability to produce antibiotics or be detrimental towards other bacteria. For the purpose of this lab, we will be testing multiple strands of bacteria. This will be done in order to observe if these strands of bacteria will show inhibition of other gram-positive and gram-negative bacteria, such as ESKAPE bacteria, which are common among hospitals. We will use the spread patch protocol to test the microbe-to-microbe interaction to identify if any antibiosis will occur.
The hypothesis will consist of two parts. The first will be that the inhibition of Bacillus subtilis and Staphylococcus aureus will occur more frequently and strongly because they are gram-positive bacteria and don't have the extra layer of lipid membrane for protection. Inhibition will not occur or occur less frequently for Escherichia coli, Serratia marcescens, Enterobacter aerogenes, and Acinetobacter bailii, as they are gram-negative bacteria that have a lipid layer for protection. The image on the right shows that the wall structure of gram-negative and gram-positive bacteria both have peptidoglycan, although the gram-negative bacteria shows to have a lipopolysaccharide outer membrane. This lipid layer decreases the permeability of the membrane, meaning it is less likely to become inhibited.
Here are a list of materials for the Antibiosis Lab. All of the materials will be used and are important for the execution of the lab procedure. After correctly labeling the plates, you will carefully dilute the ESKAPE bacteria till it reaches 0.05 McFarland standard. Be sure to dilute slowly to not alter the results. Spread all of the 6 diluted ESKAPE relatives on its corresponding plate. You will then transfer the antibiotic-producing bacteria from Lab 3 onto each ESKAPE spread plate. After incubating the plates, you will observe for ample growth, and you will then analyze inhibition growth.
In this slide, we will be looking at the initial screen results. This initial screen result tested environmental isolates against 3 bacteria, E. coli, Staphylococcus, and Bacillus subtilis. In the initial screening, the environmental isolate had the greatest inhibition against Bacillus subtilis with 179 colonies out of the total 760 that were successfully tested. About 20% of the isolates had inhibited Staphylococcus. Meanwhile, only 16% of the colonies showed activity towards E. coli. The image on the right shows successful activity against Staphylococcus aureus.
Just like the previous slide, the isolates had the most frequent inhibition of the ESKAPE bacteria Bacillus subtilis because of all the successful tested colonies shown to have some sort of inhibition, either weak or strong. Meanwhile, the isolates had the least frequent inhibition of the ESKAPE bacteria Enterobacter aerogenes because of all the successful tested colonies showed to have no inhibition.
This slide focuses on the results from the second screening. We will identify the inhibition as no reaction, weak, and strong. Weak meaning that there was less than 5 millimeters of clearing around the patch, and strong meaning that there was more than 5 millimeters of clearing. In the graph to the right, we can see that less than 20% of isolates tested on E. coli, Serratia marcescens, and Enterobacter aerogenes showed any inhibition. Over 50% of Staphylococcus aureus and Bacillus subtilis were each inhibited. Weak inhibition occurred for most of the bacteria, but not all, as there was no inhibition for Enterobacter aerogenes. Some data was not available as the plates were not completely filled with isolates. Conclusion. The purpose of this lab was to test for antibiosis from the environmental isolate against the ESKAPE bacteria. Initial screening showed high activity of inhibition with 23% of the colonies that inhibited Bacillus subtilis. E. coli showed to be resistant, as of the successful colonies, only 16% of the isolates inhibited E. coli. The data from the lab results align with our hypothesis because the gram-positive bacteria Bacillus subtilis and Staphylococcus showed to be inhibited more frequently and strongly. 50% or more of the isolates of each of these 2 plates showed activity of inhibition. The other gram-negative bacteria were inhibited less frequently. Each plate showed that less than 20% of the colonies inhibited their respective bacteria. The graph in the previous slide clearly shows a difference of inhibitions between the gram-negative and gram-positive bacteria. We predicted that there would be no strong inhibition with the gram-negative bacteria, but were proven wrong when there was strong inhibition against E. coli and Serratia marcescens.
Although this approach is simple in finding which bacteria have antibiosis activity, you can easily alter the results with a misstep of diluting the ESKAPE relatives. By diluting too much, this can result in over-inhibition, leading to antibiotic production when it normally would not occur. The same could be said when there is not enough dilution, which could result in overgrowth of the ESKAPE relative over the isolate. A solution for this would be making sure you transfer extremely small amounts of the ESKAPE relative into a saline tube and then check it with the McFarland standard constantly.